and they're not labeled. Okay, these are some of the items. So these are really common things: cereal, baby food, crackers, cake mix, bread, dog food, some of the things that you think are healthy: garden burgers, Boca burgers, Morningstar Farms burgers, Ovaltine, well, Baco's isn't very healthy, but anyway, muffin mix. All of these things had some genetically engineered food in them, crops in them. And so I was going to give you a list of these, but it's much more extensive than this. And so you can pretty much figure that most of the processed food on the shelves, unless it's certified organic and it says non-genetically engineered, whatever, has some genetically engineered food in it, food product. Okay, so breaking down the problems. The method of transferring that piece of DNA, as much as we'd like to say that it is not surgical. It's like those missiles, right? It's not surgical. When you take the piece of DNA, you're also taking bits that are on each end. So yes, you've got your target piece of DNA that codes for what you want, but you can't splice it exactly there. It's not that clean. So you've got other pieces on the sides. That also codes for stuff. Sometimes it codes for nothing. Sometimes it codes for something. And in the case where those people die, it was coding for something that actually produced a toxin within that organism. And so that DNA was unfortunately coding for a poison, and that's what was transferred into the new plant. We don't always know what it codes for until something happens. They have found side effects to some of these, death or illness from some of those pieces, and some of them are coding very toxic substances. Genes don't always code for one trait. It's not that easy. So you have some genetic information, and they code for something, yes, but they may also code for other stuff. So it's not one gene, one trait. It's actually coding for the production of proteins and for certain processes in the body of the organism. And so it's not just, make beta-carotene. It's instructions for other things as well. There may be multiple functions coded by one section. They do not necessarily code for the same thing once you put them in an entirely different organism, because there are other genes on the DNA that regulate each other. Okay? So if you splice that piece of gene in and you do not also add all the genes that regulate that gene, now you've got a gene that does not function the way it did in the original organism. You may have an uncontrolled situation because you don't have the modifying genes, and you may not have spliced those in, or they are now being directed by the genes that are in that organism to do something different. So you have the complication of taking something that was originally in one organism that functioned a certain way, putting it in another organism without adding all that extra stuff that also regulates it and makes it do exactly the same thing. Okay. The other problem is, okay, let's say that gene information isn't causing toxins, which is the first bad thing, or isn't uncontrolled or whatever. It's still in there, and it's different from what is in the wild population. And so you hear about biogenetic diversity, and you want to preserve the wild types because there is that diversity of genetics that allows for those things to evolve under different changing conditions. So now if you have a monoculture of a plant that is only of one kind, and it can actually cross with other organisms, after a while, you're only going to have that one kind. And so you're going to reduce biodiversity. So now there's controversy about whether this is happening, whether it's not. The thing is that it is spreading to the wild populations. Okay,